Hello, I'm Dong Wei. I appreciate the opportunity to be with you today. I'm here for I'm here to talk to you about kinase-mediated C1P metabolism, attenuated acute liver injury by inhibiting the KEAP1 nerve2 interaction. Our team has been dedicated to sphingolipid research. Sphingolipids are a major class of lipids present in all cells and essential to organismal development and maintenance. The metabolism pathway of sphingolipids involves the optimization of serine and palmitoyl CoA as substrates. This pathway is orchestrated by a series of metabolic enzymes in the de novo um, synthesis pathway, like the blue one, and automated leading to the production of ceramide. Ceramide is the central molecule in the sphingolipid pathway. Ceramide can then undergo further conversion into various sphingolipid molecules through different pathways, such as the salvage pathway, like the red one, and the glucose metabolism pathway, like the green one, and the sphingomyelin pathway, like the orange one. The sphingolipid pathway has been shown to have a close association with aging. In yeast, LAC1 and LAC1 were the first genes identified as important determinants of lifespan. Eventually, it was determined that the products of these genes function as thirst synthesis and were identified as longevity-associated genes. Many studies have impacted that ceramide as a key regulator of cell senescence and linked to autophagy. What's more, spingolip is involved in the cell cycle arrest, proliferation, immigration, escape of synthesis, and apoptosis, and so on. Senescence is senescence, like the feature, uh, just like the figure. Uh, Senescence is the mechanism by which cells adapted to environment stress, such as oxidative stress, oncogenes, and so on. It leads to tissues adaptation after diverse injury, but it can also result in poorly functioning cells and tissues, such as the progression of the liver disease. Various causes, like the alcohol virus and so on, cause the acute liver injury and then to the chronic liver injury and the inability of the liver to regulate nitrogen. And this can lead to cirrhosis and liver cancer. So acute liver injury is implicated to the pathologic progression of a variety of liver disease. Uh, then we found that the enzyme ceramide and C1P was upregulated in acute liver injury. C1P is protected by the ceramide kinase, and it's similar to the S1P. And it's viewed as an extension of the sphingolipid sorted. Previous studies have reported an increase in C1P level in case of micro audit. We can find that evaluated levels of C1P have been observed in the serum and the tissues, and C1P can attenuate the ovarian damage and associated with the liver oxidative stress. Since then, we induced the acute liver injury first. We found that the severity peak reached after 48 hours, as evidenced by pathologic sections and liver enzyme indicators. Cellularly, PCR reanalysis of sphingolipid metabolism revealed that a significant upregulation of SERC. We can find this upregulation both in the Western blot result and the qPCR. And then this phenomenon was further confirmed in the HII samples. Then treatment with the SERC inhibitor NVP 231 in vivo will exacerbate acute liver injury, leading to earlier peak of the liver enzyme and the bigger area of the apoptosis, which indicates that a protective role of SERC against liver damage. What's more, addition of C1P or SERC inhibition or knockdown in oxidative damage model of hepatocyte further confirm that the protective role of SERC in liver cells. We can find the when inhibition or knockdown the SERC will reduce the cell viability and the more ROS. After elucidated the roles of SERC and C1P, we want to know how C1P works. So we identified the NERV2 and the KEAP1, the key players in oxidative damage, and observed a significant alteration only in the protein expression of NERV2. Especially, we observed an upregulation of NERV2 protein levels following the expression inhibition of SERC. Interestingly, the RNA level of NERV2 and the protein RNA level of KEAP1 did not show consistent changes. This led us to hypothesize that C1P might influence the interaction of nerve2 and the KEAP1, but not the expression of KEAP1. Since the nerve2, uh, since when blocking the binding of nerve2 and KEAP1, the KEAP1 and the nerve2 is kept from the KEAP1's degradation, and the nerve2 translocated to the nucleus, and its protein level will upregulated, but there is no significant regulation on the RNA level and KEAP1. So our research found that C1P does indeed impacted the interaction between nerve2 and KEAP1, as demonstrated through various techniques such as the nuclear cytoplasm separation and co-localization and pull-down experiments. In our investigation into how C1P influence the binding nerve2 KEAP1, we observe that C1P especially binds to KEAP1, but not nerve2. Through molecular docking analysis, we hypothesize that C1P interacts with the DGR domain of KEAP1, which is the binding areas of the KEAP1 to the nerve2. This hypothesis is supposed that by the experiment involving the expression of KEAP1 proteins with different domain deletions, such as the 5 domain deletions. Only when the DGR domain deletion, the KEAP1 protein can't binding to the C1P bits. So it confirms that the binding city of C1P is indeed located within the DGR domain. In the absence of oxidative stress, two KEAP1 proteins acting as subtracted adapter proteins of E3 ubiquin ligands form homodimers. The DGR domains of these KEAP1 proteins then bind to nerve2, leading to the targeting of nerve2 for ubiquitination and degradation in the proteasome. However, when oxidative stress occurs, there is a compensatory increase in SERC expression. This increases the SERC activity, resulting the production of C1P, which binds to the DGR domain of KEAP1. This band interferes with the interaction between KEAP1 and nerve2, causing nerve2 to dissociate from KEAP1. As a result, nerve2 escaped KEAP1 mediated degradation and translocates into the nucleus, where it promotes the prescription of antioxidant genes. Well, that's about it for now. sex.